Hello guys, welcome to a new video. Today we'll be discussing about urea cycle. In the last session we have seen that ammonia is released from amino acids by deamination process, and also we have seen that this ammonia released is very toxic for our body. In this session we'll be discussing about the conversion of ammonia or detoxification of ammonia to urea. So how this happens? Actually, ammonia from all over our body reaches the liver. In the liver, it gets detoxified to urea. Then, this urea gets excreted through our kidney. This is how ammonia gets detoxified to urea. This is just a general outline of the detoxification. Now, let us see in detail what urea cycle is. So, what is urea cycle? Urea cycle is a biochemical pathway in our body where ammonia gets converted to urea. It was first introduced by Hans Krebs and Kurt Hansleit in 1932. So after the names, this cycle is also called as Krebs-Hansleit cycle. This cycle is also known as ornithine cycle because ornithine is the first member of this cycle. All the reactions of this cycle occurs in our liver, and there is a total of 5 reactions in this cycle, and among it the first 2 reactions occurs in the mitochondria of liver, and the next 3 reactions occurs in the cytosol of our liver. The enzymes of the first 2 reactions are concentrated in the mitochondria, and the enzymes of the next 3 reactions are concentrated in the cytosol of liver. So it takes place in the two places, that is, first 2 reactions occurs in mitochondria and the next 3 reactions occurs in the cytosol of liver. Now let us look on to the 5 steps of urea cycle. The first step is the synthesis of carbamoyl phosphate. Second step is the synthesis or formation of citrulline. Third step is the formation of argininosuccinate. Fourth step is the synthesis or formation of arginine. And the last step is the conversion of arginine to urea. The first 2 reactions of urea cycle, that is the synthesis of carbamoyl phosphate and the synthesis of citrulline, occurs in the mitochondria of liver. The next 3 reactions, that is the formation of argininosuccinate, the formation of arginine, and the conversion of arginine to urea, occurs in the cytosol of the liver. So these are the 5 steps of urea cycle, and the first 2 reactions occurs in the mitochondria and the next 3 reactions occurs in the cytosol. Now, let us look into these reactions in detail. So the first reaction is the synthesis of carbamoyl phosphate. This reaction takes place in the mitochondria of liver in presence of carbamoyl phosphate synthase 1 or CPS1 enzyme. This reaction involves the coupling of an ammonia molecule with a bicarbonate ion to form carbamoyl phosphate. If we look here, we can see that the bicarbonate ion reacts with an ATP molecule to form carboxyphosphate. The phosphate group that is released from ATP has been joined with the oxygen of bicarbonate, and hence the carboxyphosphate is formed. Next, the carboxyphosphate react with ammonia to form carbamic acid. The NH2 group of ammonia replaces phosphate in the carboxyphosphate and forms carbamic acid. So we can see here an NH2 molecule in place of phosphate group. Next, the carbamic acid then reacts with ATP to form carbamyl phosphate. Thus, we can say that CO2 of bicarbonate, NH2 of ammonia and phosphate group of ATP join together to form carbamoyl phosphate in presence of CPS1 enzyme. This enzyme requires N-acetyl glutamate for its activity as a cofactor. So this is the first step of urea cycle, that is the formation of carbamoyl phosphate. So let us move on to the next reaction, that is the synthesis of citrulline. As I said earlier, this reaction is also mitochondrial because the enzymes for this reaction are distributed in the mitochondria of liver. Here, the carbamoyl portion of carbamoyl phosphate is transferred to ornithine, which is an amino acid which present in mitochondria and result in the formation of citrulline. This reaction takes place in presence of enzyme ornithine transcarbamylase with the release of inorganic phosphate. If you look here, this is the carbamoyl group of carbamoyl phosphate, and this group react with the amino group of ornithine to form citrulline. The citrulline thus formed is transported to the cytosol of liver for further reactions. So we have seen the first two mitochondrial reactions of urea cycle. Now let us move on to the third reaction, which is a cytosolic reaction. So the third reaction is the formation of arginosuccinate. Here, citrulline, which has been transported from mitochondria to cytosol, react with aspartate to form arginosuccinate. The enzyme which catalyzes this reaction is the arginosuccinate synthetase. Here, the NH2 group or amino group of aspartate react with the carbon donated by the carbamoyl phosphate to form arginosuccinate. For this reaction, two high energy bonds of ATP has been spent for provision of energy. So this is the third step, which is the formation of arginosuccinate. Let us move on to the fourth step, that is the formation of arginine or the cleavage of arginosuccinate. Here, the arginosuccinate, which is formed in the previous reaction, is cleaved to form arginine and fumarate in presence of arginosuccinase enzyme. This enzyme is also called as arginosuccinate lyase enzyme. If we look here, we can see that this bond between nitrogen and carbon is broken, and one of the hydrogen from this carbon has been donated to this nitrogen to form arginine, and the leftover carbon skeleton has formed fumarate. So the arginine formed here serves as an immediate precursor of urea. The fumarate produced enters the TCA cycle for further reactions. So this is the formation of arginine from arginosuccinate. That is the fourth reaction. Now let us go on to the last and final reaction of urea cycle, that is the conversion of arginine to urea. This reaction takes place in presence of arginase enzyme. If we look here, we can see that this bond between this carbon and this nitrogen is broken, and the oxygen of water comes and binds with this carbon, and one of the hydrogen of water comes and binds with this NH group and forms urea. The remaining hydrogen of water comes and joins with this NH group and forms ornithine. We can see here that ornithine, which was the first member of urea cycle, is regenerated here, and it is transported back to mitochondria to begin another cycle, and urea, as we all know, is excreted. So this is how urea is generated from ammonia. This is what urea cycle is. Finally, we can say what urea cycle is. It is a biochemical pathway which involves the conversion of ammonia to urea through a series of 5 reactions. So this is all about this cycle. After the synthesis of urea, it diffuses from liver and is transported in blood to kidneys. In the kidneys, this urea is filtered and excreted in urine. This is how urea becomes an excretory product. Now, let us look on to the sources of atoms in urea. Here, one of the nitrogen groups are donated by ammonia, and the second nitrogen group is donated by amino acid aspartate, and the carbon and oxygen groups are donated by the carbon dioxide molecule. Now, we will move on to the significance of this cycle. First of all, it helps in the toxic ammonia conversion to urea. That is, the toxic ammonia is converted to non-toxic urea. Second one is the disposal of waste products such as ammonia and carbon dioxide. And third one is the synthesis of essential amino acids such as arginine. Now, let us move on to the disorders caused by deficiency of enzymes involved in urea cycle. The first disorder is hyperammonemia type 1. It is caused by the deficiency of enzyme carbamoyl phosphate synthase 1, and due to its deficiency, ammonia gets accumulated. and due to the deficiency of the second enzyme ornithine transcarbamylase, ammonia gets accumulated and causes hyperammonemia type 2. Due to the deficiency of the third enzyme argininosuccinate synthase, citrulline gets accumulated and causes citrullinemia. Due to the deficiency of argininosuccinase enzyme, argininosuccinate gets accumulated and causes argininosuccinic aciduria. Due to the deficiency of the final enzyme arginase, arginine gets accumulated and causes the disorder argininemia. So these are the disorders due to deficiency of enzymes in urea cycle. So this is all about urea cycle. We have seen the different reactions, its sources, its significance, its disorders. So thank you for watching. Please subscribe, like and share. In the next video, we will see the individual amino acid metabolisms.